In the cold water far below the reach of any sun, a single molecule meets an enzyme, and the meeting releases light. No heat comes with it, no sound, only a photon arriving in the dark as if the dark had briefly remembered something, and then it is gone. The molecule that made it has changed its shape. The enzyme that helped it is ready to help again, and in the surrounding water, which has been cold and black for longer than there have been creatures to notice, the event leaves no trace at all. This is where the story begins, not with a creature, not with a vast and alien ocean, with a molecule and what happens to it when oxygen arrives, and the particular quality of the light that results, cold, brief, and exact. The molecule is called luciferin. The enzyme is called luciferase. Together, their interaction produces something that living tissue almost never produces: visible light, released directly from a chemical reaction, without any fire, without any filament, without any of the heat that every other light source in ordinary experience produces alongside its glow. The light that comes from a flame comes wrapped in heat. The light from a bulb passes through a wire so hot it glows, but the light from a living organism, from the reaction of luciferin and luciferase in the presence of oxygen, costs almost nothing in warmth at all. Scientists call it cold light, and the name is precise. In a tissue already a few degrees above freezing, in water that has never been warmed by the sun, the reaction produces a photon and very nearly nothing else. What luciferin actually is, at the chemical level, is a relatively small organic molecule whose structure allows it to absorb oxygen in a way that lifts some of its electrons to a higher energy state. Those electrons do not stay there, they fall back. And as they fall, the energy they release does not become heat, but becomes light instead, a single photon whose wavelength falls most often in the blue-green range of the visible spectrum, between roughly 450 and 500 nanometers. Blue-green is what the deep sea transmits best. Other wavelengths are absorbed by seawater long before they travel far. Blue and green travel deepest, and so it is blue and green that evolution has found, again and again, in the light-producing chemistry of ocean organisms. The photon that leaves the luciferin molecule is already tuned by the physics of water to the color most likely to reach a distant eye. Luciferase is the enzyme that makes the reaction happen at a useful rate. Without it, luciferin would still oxidize, but slowly, too slowly to produce a flash, too diffuse to signal anything, too faint to serve any purpose. With luciferase, the reaction accelerates. The enzyme binds the luciferin molecule, holds it in the right configuration, brings it into contact with oxygen in a precise and repeatable geometry, and the result is a controlled release, a flash, a glow, a steady emission, depending on how the tissue is built and how the organism has arranged the molecules within it. The enzyme does not change in the reaction. It is released intact, ready to receive another luciferin molecule, and the process can repeat as long as both ingredients are present. In the tissue of a living creature, this means the light can be sustained for as long as the organism chooses to sustain it, held open, pulsed, switched off, because the only things required are the molecule, the enzyme, and oxygen, and all three are available as long as the organism is alive and regulating them. The reaction is known, the chemistry is understood, and yet the first thing to know about bioluminescence, the first thing that shapes everything else about it, is that this chemistry did not arise once in the history of life and then spread. It arose many times, in many forms, in many kinds of organism, across hundreds of millions of years of separate evolution. The light that a firefly produces above a summer field uses a luciferin that is chemically distinct from the luciferin that glows inside a marine worm. The glow of a dinoflagellate in a breaking wave uses a different molecule still. A fish living in the permanent dark of the deep sea carries yet another variant, evolved entirely independently of the others. The word luciferin is not the name of a single molecule. It is a name given to a whole category of structurally different compounds that all happen to do the same thing. They oxidize in the presence of their partner enzyme, and they release the energy as light. There are at least 8 chemically distinct forms of luciferin known to exist across nature. They do not share the same molecular structure. They were not inherited from a common ancestor. They were arrived at separately, each one a different solution to the same problem posed by chemistry and darkness. The most widespread of these forms, found in more marine organisms than any other, is a molecule called coenterasine. It is found in jellyfish and squid and crustaceans and fish and radiolarians and a number of other ocean creatures distributed across enormous evolutionary distances from one another. The fact that so many unrelated organisms carry the same molecular form of luciferin is partly explained by the way the ocean works. Coenterasine is present in marine food webs, and animals that eat other animals that produce it can accumulate it in their own tissues without synthesizing it themselves. Some marine organisms appear to obtain their coenterasine through diet rather than through their own biosynthesis. The molecule circulates through the ocean's food web the way other biochemical resources do. But the luciferases that react with coenterasine in different species are not the same. The enzymes are different proteins built from different gene sequences shaped by different evolutionary histories, and they share no meaningful similarity at the level of their molecular structure. 2 organisms can carry the same luciferin and use entirely different enzymes to light it. The substrate is shared. The machinery is not. This matters because it is the luciferase gene that carries the clearest record of how bioluminescence evolved. When scientists compare the genetic sequences encoding luciferases in different marine species, fish and squid, and bacteria and worms, and look for shared ancestry in those sequences, they find none. The genes have no common origin. They were built separately, by evolution, from whatever protein-coding genetic material was available to each lineage at the time. In many cases, the ancestral proteins from which luciferases were co-opted appear to have been enzymes already involved in the management of fatty acids, proteins whose job in the cell was originally something else entirely, something to do with the metabolism of fats, that were gradually reshaped by natural selection into something capable of catalyzing the light-producing reaction. The change from a fatty acid oxidase to a luciferase did not require a radical reinvention. It required an incremental shift in what the enzyme bound, and how, and what happened to the electrons when the reaction completed. Given enough time and enough small genetic changes, the same class of chemical reaction can be redirected from one product to another. And in dozens of separate lineages across the history of life, that redirection appears to have happened, not because any organism was seeking light, but because the variants that produced a little light, or that could control a little light, survived and reproduced slightly better than those that did not. Bioluminescence has evolved independently, by the most careful current estimates, at least 40 to 50 times across the full tree of life. This is a number that bears sitting with for a moment. Not 40 to 50 species with bioluminescence, not 40 to 50 populations, 40 to 50 separate evolutionary origins, 40 to 50 times that a lineage of organisms, facing the particular pressures of their environment, arrived at the same biochemical solution through a completely independent path. The genetic evidence makes this clear because if bioluminescence had a single common ancestor, the genes encoding it would share a common structure across all the organisms that carry it, the way the genes for producing hemoglobin or for managing photosynthesis share common ancestry across the organisms that carry those. The luciferase genes do not share that ancestry. Each one is its own story, its own evolutionary path, its own particular reshaping of whatever raw material the lineage had available. In fish alone, molecular phylogenetic analysis, the careful comparison of genetic sequences to reconstruct evolutionary relationships, suggests that bioluminescence evolved independently at a minimum of 27 separate times. 27 times within a single major group of vertebrates. 27 times in creatures with backbones and jaws and fins and eyes, organisms that share a great deal of their biology with one another, but nevertheless arrived at the production of light through 27 entirely distinct evolutionary paths. Fish that glow on their bellies, and fish that glow on their chins, and fish that carry lights on long modified spines, and fish whose whole skin can emit a faint steady blue, and behind each of these a separate molecular history, a separate ancestral protein that was gradually reshaped by time. The scale of this repetition is worth feeling carefully. In evolutionary biology, convergent evolution, the independent arrival of different lineages at the same solution, is not unusual. Eyes evolved independently in vertebrates and mollusks and arthropods. Wings evolved independently in birds, bats, and insects. Warm-bloodedness evolved independently in mammals and in some fish. These are well-documented cases of convergence, each one the result of a particular adaptive problem that had a particular physical solution, and different lineages that face the same problem, finding the same solution without inheriting it from a common ancestor. But bioluminescence, with its 40 to 50 independent origins, stands as one of the most extreme cases of convergent evolution in all of biology. The problem, produce light without heat in a dark environment, had such a clear and available chemical solution that nature found it not once, not a handful of times, but dozens of times across hundreds of millions of years. What the chemistry requires is not elaborate. A relatively small organic molecule capable of oxidation, an enzyme that binds it and speeds the reaction, oxygen. These are ingredients that living chemistry, broadly, already works with. The baseline of organic metabolism involves oxidation reactions, oxygen transport, enzyme-catalyzed chemistry. All the raw materials for bioluminescence are things that cells were already doing in some form before any of them produced light. The step from ordinary oxidative chemistry to light-producing oxidative chemistry, from the perspective of evolution, is a small step. It does not require a wholly new category of biochemistry. It requires a modification of existing chemistry. And when a small modification is all that is required, and when that modification provides even a slight advantage to the organisms that carry it, evolution finds it repeatedly because the path is short and the reward is real. There is a version of this story in which bioluminescence is presented as a deep puzzle, a wonder that defies explanation. Why would life produce light, as though light in the dark were an extravagance? But the chemistry argues the opposite. Given the materials that living cells already handle, given the environments that many marine organisms inhabit, given the uses that a controlled light source has in a world of perpetual darkness, the question is almost reversed. Given how accessible the chemistry was, given how accessible the chemistry was, given how accessible the chemistry was, and how many times it was found, Why would any organism that spends its life in the dark not eventually arrive at light? In the Mesopelagic Ocean, the zone of water between roughly 600 and 3,000 feet below the surface, where sunlight fades to nothing and the world becomes permanently dark, the density of bioluminescent organisms is not the exception. It approaches the rule. In the open water of this zone, surveys have found that the great majority of the living creatures present, measured by count, not by mass, produce light in some form. The ocean below the reach of the sun is not mostly dark. It is populated by organisms that carry their own illumination, that use it for the full range of purposes that light serves for any animal: to be seen by a mate, to hide from a predator, to attract prey, to signal an alarm, to navigate the permanent night. But all of that, the purposes and the uses and the particular creatures and their behaviors, comes later. At this level, the question is simpler. What is bioluminescence at the level of the molecule? And the answer is a cold release of light from an oxidation reaction carried out by a substrate and an enzyme in the presence of oxygen, resulting in a photon whose color is tuned by physics to the wavelength that travels farthest through seawater. A luciferin molecule, before it is oxidized, holds the energy that the light will carry. The electrons in its structure exist in a particular arrangement, a particular distribution around the atoms of the molecule, and that arrangement is stable, not reaching for anything, not tending toward change on its own. When luciferase brings oxygen into contact with the molecule in the right configuration, the arrangement changes, the electrons shift. The molecule is oxidized, it gives up electrons to the oxygen, and in the process, the energy that was stored in that particular electron arrangement is released. In most oxidation reactions in biology, that energy becomes heat, or it is captured in the bonds of other molecules, like ATP, to be used later. In the luciferin-luciferase reaction, the energy does neither of those things. It goes directly into lifting electrons to a briefly excited state, and when those electrons fall back to their resting arrangement, the energy comes out as a photon of light. The transition is direct. Heat is a form of disordered molecular motion, and the luciferin reaction does not produce that disorder in the normal quantity. The energy goes to light instead, with very little wasted on the warm, disorganized molecular collisions that produce heat in other reactions. This is what is meant by cold light. Not that the light itself is cold, but that it is produced without the byproduct of heat that almost every other light-producing process generates. A candle is hot, an incandescent bulb is hot enough to burn. The bioluminescent tissue of a jellyfish producing light in the cold water of the Pacific Ocean is the same temperature before, during, and after the reaction. The light leaves, the tissue is unchanged, and the surrounding water, already near freezing, is no warmer for having been illuminated. There is something clarifying about that. The light serves only as light. It is not a byproduct of warmth, not the visible edge of a process whose real purpose is heat. It is produced deliberately, in the sense that every evolved structure is the result of selection for the outcomes it produces. It is issued cleanly, carrying the energy it carries, and the organism that produces it does not pay a thermal cost for doing so. In environments where energy is scarce, as the deep ocean nearly always is, this efficiency is part of what makes bioluminescence viable as a sustained biological tool. The organism is not burning calories to stay warm in order to make light. The light costs what the chemistry costs, and almost nothing extra. Coilenterazine, the form of luciferin most common in marine organisms, is a molecule that has been found in species representing more than 100 distinct animal families. It appears in the tissues of jellyfish, and of the fish that eat jellyfish, and of the larger fish that eat those fish. It cycles through the ocean's food web the way nutrients do, passing from organism to organism through predation and digestion. Some animals that carry it seem capable of synthesizing it themselves. Others appear to acquire it entirely through diet, incorporating it into their photophores, their light-producing organs, from the tissues of what they eat. The molecule is, in this sense, a biochemical resource of the deep ocean, something the water and the creatures in it have been managing and cycling and using for an enormous span of time. When scientists first began characterizing it in the 20th century, they found it in so many unrelated organisms that its widespread distribution became one of the early indications of how differently bioluminescence had evolved in different lineages, not as an inherited trait passed down from a single common ancestor, but as a chemical resource independently harnessed by many different evolutionary lineages that each developed their own enzyme to use it. The luciferases paired with coelenterazine are each distinct. In the crystal jelly, a translucent organism that drifts along the Pacific coast, the enzyme involved is not a luciferase in the traditional sense, but a photoprotein, a different kind of structure, one that binds the luciferin molecule and oxygen together in a stable complex, and releases the light only when triggered by a specific signal, in this case the presence of calcium ions. The light is held, in a sense ready in the protein, waiting for the cellular signal that will release it. In copepods, small crustaceans that fill the ocean's open water, the luciferases are different proteins again, sharing no meaningful structural resemblance to the photoprotein of the jellyfish, or to the luciferases of the fish, or to the enzymes found in bioluminescent bacteria. Each lineage arrived at its own solution. Each enzyme is a separate piece of molecular engineering, built from the genetic raw material that particular lineage had available, refined over whatever period of time was required for selection to shape it into something that produced enough light to make a difference. 8 distinct forms of luciferin, dozens of distinct luciferases, 40 to 50 independent evolutionary origins. These numbers, taken together, describe not a single biochemical invention, but a recurring discovery, the same fundamental possibility, latent in the chemistry of oxygen and organic molecules, found again and again by organisms that had no connection to one another, beyond the shared medium of the ocean and the shared fact of living in the dark. There is a useful way to think about this. Evolution does not plan, does not look ahead, does not select for traits because they will be useful in the future. It selects for traits because they increase reproductive success right now, in the current environment, with the current pressures and the current resources. Every independent origin of bioluminescence represents a moment stretched across thousands of generations, compressed in the logic of natural selection, when the chemistry that produced a little light became, in a particular place and time, a trait worth having, not because light is beautiful, or because darkness is frightening, or because any organism had a preference for illumination. Because the organisms that produced a little light in the right circumstances passed on more copies of themselves, and the organisms that produced a bit more light, more controllably, with a bit more variation in how and when and where they produced it, passed on still more. And over time, in lineage after lineage, in ocean after ocean, in depth after depth, and era after era, the cold chemical reaction of luciferin and luciferase became something that millions of species carried in their cells. The deep sea is full of this light, not the reflected light of the sun, which does not reach below a certain depth, not the filtered light of a moon or a lamp, light that comes from within living tissue from the controlled oxidation of small organic molecules produced without heat, emitting a photon at a wavelength the ocean can carry, visible to the eyes of creatures evolved to receive it. Light that has been produced in the deep ocean since before the Cambrian period. Some evidence suggests since before the great diversification of complex animal body plans that the fossil record documents beginning around 540 million years ago. The chemistry predates the creatures. The reaction was possible, in the general sense, as soon as organisms existed that managed oxygen and oxidative metabolism and organic molecules of sufficient complexity. Evolution found it early, it found it often, and it kept finding it in lineage after lineage, because the dark ocean kept posing the same questions to whatever lived inside it, and the chemistry of light kept being one of the answers. None of this is magic. It is something more considered than magic, more patient, more exact. It is chemistry, a specific molecule, meaning a specific enzyme in a specific geometry, releasing a specific form of energy at a specific wavelength, and it is evolution, the same chemistry being found independently dozens of times across the widest reaches of the tree of life, each time shaped slightly differently by the history of the lineage that found it, each time serving slightly different purposes in slightly different bodies, in slightly different depths of water. The precision of the chemistry and the scale of the evolutionary repetition together describe something that resists easy characterization. It is not rare, it is not unique, it is in a sense almost inevitable, the logical outcome of placing oxygen-managing organic chemistry in a dark environment and allowing hundreds of millions of years for natural selection to work. And yet, a single luciferin molecule, oxidized by its enzyme in cold water in the deep ocean, releasing one photon of blue-green light into the absolute dark. This is not diminished by being understood. The photon crosses the water at the speed of light. If there is an eye to receive it, the eye receives it. If there is no eye, the photon is absorbed by the water, which warms by an amount so vanishingly small it cannot be measured. The event is complete in itself. The chemistry is exact. The dark around it is exactly as dark as before, except for the one moment during which it was not. Somewhere between 200 million and 500 million years ago, the estimates vary, and the fossil record for soft-bodied organisms in the deep sea is sparse. Some organism in some ocean carried a mutation that changed how one of its proteins folded and what molecules that protein bound, and what happened to the energy when the reaction completed. The change was small, the change was chemical, and the result propagated through that organism's descendants across all the time since has been living light, cold, exact, and blue-green, repeated in 40 to 50 separate evolutionary lines, carried into every dark habitat in the ocean, found by evolution the way water finds a low place, not because any force was directing it there, but because the path was available, and the chemistry was ready, and the dark kept being dark. Imagine the evolutionary tree of life, that vast branching structure representing every species that has ever lived, rooted in the first cells and spreading outward across 4 billion years of time. Along its limbs, organisms diverge and adapt and sometimes go extinct and sometimes persist unchanged for millions of years. The limbs branch into smaller branches and those into smaller ones still, until the outermost twigs represent individual species alive today. Now imagine small lights, each one a cold blue-green point, appearing on that tree, not gathered in one place, not inherited from a single node, and carried down through the branches, scattered instead across 40 or 50 separate places on the tree, each light appearing independently on a limb where no light appeared before, the result of its own particular evolutionary path in its own particular lineage. Some of the lights are close together in time. Some are separated by hundreds of millions of years. Some are on lineages that share almost nothing else in common: a bacterium and a fish and a jellyfish and a squid, each appearing on a different part of the tree, each carrying the same cold chemistry arrived at by a different root. That is what the genetic evidence shows. That is what the comparison of luciferase sequences reveals when they are laid side by side and found to share no common origin. That is what the 40 to 50 independent evolutionary origins of bioluminescence mean, translated from a number into a picture. The same reaction, found again and again across the full width and depth of life's history, because the chemistry was available and the dark was vast, and the organisms inside that dark kept finding, generation by generation, that the ones who could make a little light did a little better, and so the light persisted and spread and multiplied and is still there in the cold water, far below the reach of any sun. A molecule meeting an enzyme and releasing something the ocean is very good at carrying. In a world where living light has been arriving independently from dozens of directions at once for longer than there have been eyes to see it. At 200 meters below the surface of the open ocean, the last measurable sunlight ends. It does not end all at once. It thins, meter by meter, the way color drains from a bruise, the red wavelengths gone first, absorbed within the top few feet of water, then the oranges, then the yellows, each drawn out of the spectrum in order as the depth increases and the water above thickens. By the time a diver descends to 60 feet, the world is already blue. By 100 meters, it is blue-gray. By 200, the blue itself has thinned to something closer to the memory of color than color itself, a faint, directionless luminosity that is less a light than the last diminishing echo of one. And then, somewhere in those deepest reachable meters of the twilight, it is gone. Not dim, not dark in the way a room is dark when someone turns off a lamp. Gone in the way that a thing can only be gone when it was never meant to be there in the first place. Below that boundary, the water is black in a way the surface world has no good word for. This transition, the boundary between lit water and dark water, sits at roughly 200 meters and has a name. It marks the upper edge of what oceanographers call the mesopelagic zone, sometimes called the twilight zone, a name that holds a small truth inside it. Twilight is a time, not a place. It is the brief period between one condition and another, the threshold state where something is ending but has not quite ended. The mesopelagic is a place that feels like that. Its upper boundary still carries a trace of the surface, a faint blue-green scatter of photons that have traveled a long way down and are nearly spent. Its lower boundary, at around 1,000 meters, gives onto something with no ambiguity at all. Between those depths, life crowds into the gradient, hovering, migrating, feeding in the half-light, using whatever faint illumination remains to see by, to hide by, to navigate the narrow overlap between the world above and the world below. The gradient is steep. At the upper edge of the mesopelagic, incoming sunlight measures roughly 1% of what reaches the surface. 1% is not nothing. 1% of full tropical noon is still a usable signal, still enough to throw a faint silhouette, still enough for certain eyes to detect. But that 1% is the ceiling of the zone, the most light this layer ever receives, and below it, the light falls steeply. By 500 meters, what remains is a fraction of a fraction. By 800 meters, measurements approach zero. The photons arriving at 900 meters have been traveling through water for so long, and through so much of it, that they are isolated, individual, scattered, arriving by chance rather than by beam. At 1,000 meters, the last of them are gone. The water below 1,000 meters is called the bathypelagic zone, and it is the largest habitable space on the surface of this planet by volume. It extends from 1,000 meters to roughly 4,000 meters in depth, a vast column of cold water that circles the Earth beneath every ocean, beneath every sea. The water there is dark in every direction. No photon of sunlight has ever reached it. No sunlight ever will, not because of any failure in the physics of light, but simply because of distance and the absorptive character of water. The 200 meters of ocean above represents more optical thickness than the entire atmosphere above it does to the light coming in from space. To sink 1,000 meters into the sea is to pass through something more opaque than all the air above you, stacked and doubled. The temperature of the bathypelagic water sits between roughly 2 and 4 degrees Celsius. This is not a temperature that varies much. It is not a temperature that changed much during the last ice age or during the warm, shallow sea periods of the deep past. The water in the midnight zone has been cold for so long that calling it cold feels insufficient. It is closer to saying that a stone wall is hard. The coldness of this water is structural. It is old. It is geological in the way it sits, stable, barely stirring, the same from season to season and from century to century in any way the body could measure. At the surface, the ocean breathes with the seasons, warming in the summer hemisphere, cooling when the angle of the sun changes, mixing in storms, stratifying in calms. Below 1,000 meters, none of that reaches. The mixing stops. The water becomes its own steady state. A degree or two of cold is not the most demanding physical fact of the midnight zone. The pressure is. At 1,000 meters, the weight of the water column above presses inward at approximately 100 atmospheres. 1 atmosphere is the weight of the entire air column above sea level, the pressure that a human body is built for, the pressure the body barely notices because it has lived inside it since birth. 100 atmospheres is 100 times that. It is not a pressure the human body can survive without extraordinary engineering to contain it, and even then the engineering must be exact. For every 10 additional meters descended from 1,000, the pressure increases by another atmosphere. At 2,000 meters, the pressure is 200 atmospheres. At 4,000 meters, it reaches 400. These numbers are precise, and they are also nearly impossible to hold as felt experience. There is no bodily analogy that scales. The closest available language is structural. A human lung, unprotected, would be crushed flat in the upper mesopelagic. The organs that evolved in surface air are not wrong. Exactly. They are simply shaped for a different column of gas. The animals of the midnight zone are not shaped for a different column. They are shaped for exactly this one. Their proteins, the folded molecular machines that carry out every function of a living cell, have evolved configurations that remain stable under this weight. In many of these organisms, a small molecule called trimethylamine oxide accumulates in tissues in concentrations that scale with depth. This molecule counteracts the effect of pressure on protein structure, stabilizing the folds that pressure tends to disrupt, functioning as a kind of molecular cushion between the organism and the physics of its environment. The deeper a species lives, the higher the concentration found in its tissues. It is not a solution the fish discovered, or worked out, or chose. It is a solution that accumulated in populations of animals living at various depths over millions of generations, because the individuals who happened to have slightly higher concentrations of it survived slightly better and passed their chemistry on. The result is a fish that carries the deep sea inside it at the molecular level. Deep in the water column between the fading of the surface light and the permanent dark below it, Another kind of movement happens quietly, continuously, and has been happening for as long as there have been organisms in the sea above. Organic matter dies at the surface. Phytoplankton, small animals, the shed skins of crustaceans, the fecal pellets of zooplankton, all of it, in the normal and ongoing course of oceanic life, sinks. It sinks slowly. Individual particles are small and light, and the water resists them, and they drift as much as they descend, moving with the currents that shift through the mesopelagic, while gravity draws them, grain by grain and flake by flake, toward the floor. This slow, continuous downward drift has a name. It is called marine snow, for the way it looks through an underwater light source. White flecks against black water, drifting in no hurry towards somewhere very far below. Marine snow is food. It is nearly the only food that arrives in the midnight zone from above. The organisms that live in the bathypelagic zone cannot photosynthesize. They cannot reach the surface. Their world is chemically poor in the way that a room with no kitchen and no supply line is poor. The resources that sustain life are not generated here. They drift down from somewhere else in particles small enough that a single falling flake might be a meaningful meal for a small creature positioned in its path. The flux of marine snow varies. It is higher in productive ocean regions where the surface waters bloom with life each spring and summer, and lower in the great gyres where the surface is almost as barren as the deep. But it never stops entirely. Across every ocean, through every season, the slow descent of the dead surface world drifts down through the dark to feed the living world below. It falls through water that barely moves. The deep currents that circulate through the bathypelagic are real and persistent. Thermohaline circulation carries cold, dense water across ocean basins over centuries, but at any local scale, in any given column of water, the movement is gentle. The water does not storm. It does not churn. It drifts in slow, cold, mineral-dense flows that take decades or centuries to complete a circuit. In this stillness, a flake of marine snow can take weeks to reach the bottom from where it was produced at the surface. 1,000 meters is not a great distance by surface standards. It is less than a mile, but in the resisting medium of cold seawater, with only the gentlest gravitational pull on a nearly neutrally buoyant particle, the descent is long. The flake may be eaten partway down. It may be broken apart by bacteria and remineralized before it ever reaches the floor. What arrives at the seafloor is often only a fraction of what set out from above. What arrives is, even so, enough to sustain communities of extraordinary diversity and patience. The floor of the deep ocean is not uniform. Over the broad reaches of the abyssal plain, the flat, sediment-covered basins that cover more of Earth's surface than any other single landform. The seafloor is a quiet place. Soft sediment accumulates over millennia. Sea cucumbers move across it so slowly that they leave trails in the silt that persist for days. Sponges filter feed from the same position for what may be decades. The pace of life on the abyssal plain is calibrated to scarcity. Food arrives slowly, and organisms here have evolved metabolisms to match, slow, economical, patient, in a way that has no emotional content, only the biological logic of a system shaped to last on almost nothing. But the deep sea floor is not only plain, where the tectonic plates that make up the ocean floor spread apart. The crust cracks. The crack is not dramatic, not sudden. It is the slow expression of convection currents deep in the planet's interior, the steady upwelling of heat from below, pushing the plates apart at rates that match the growth of a fingernail. Where the crack opens, seawater filters down into the heated rock and reemerges as superheated mineral-rich fluid, carrying dissolved metals and sulfur compounds that the surrounding cold ocean water does not contain. These exits are called hydrothermal vents, and they push water that has been heated to several hundred degrees Celsius out through chimneys of precipitated minerals, while the surrounding ocean sits near 2 degrees. The temperature differential at the vent opening from the superheated fluid to the near freezing ocean is one of the sharpest thermal gradients on the planet. It is not a place that looks livable from the outside. From the inside, it is teeming. The first time a human being laid eyes on a hydrothermal vent, it was 1977. The research submersible Alvin had been directed to investigate thermal anomalies along the Galapagos Rift, a spreading center in the eastern Pacific Ocean, at a depth of about 2,500 meters. What the scientists aboard saw when they arrived was not what any biological expectation had prepared them for. Around the vent openings, in the total absence of sunlight at the bottom of the ocean, there were animals. Dense communities of animals packed around the vent structures in a concentration the surrounding seafloor never shows. Tube worms in clusters, their red plumes waving in the warm current rising from the vent. Clams the size of dinner plates. White crabs moving through water that minutes before had been too hot to touch. Life, concentrated and apparently thriving, at a place and depth where life was expected to be minimal. What sustained them was not the sun. On the surface of the ocean and in the sunlit shallows, nearly all food webs begin with photosynthesis. A photon of sunlight strikes a chlorophyll molecule in a phytoplankton cell, and the energy of that photon drives the conversion of carbon dioxide and water into sugar. The sugar feeds the phytoplankton. Something eats the phytoplankton. Something eats that. The chain runs from the photon all the way up to the whale, and if the light stops, the food stops. The entire sunlit ocean is at base a light-powered system. At the hydrothermal vent, the sunlight has been gone for 2.5 kilometers of water. None of the energy reaching the vent community came from above. It came from inside the planet. The fluid emerging from the vents carries chemical energy in the form of hydrogen sulfide, and certain bacteria have evolved the metabolic machinery to harvest that chemical energy directly, to use hydrogen sulfide the way surface plants use sunlight, as a fuel for fixing carbon and building organic matter. This is chemosynthesis. It is not photosynthesis with different ingredients. It is a separate solution to the same fundamental problem, which is where to get energy. Chemosynthesis does not require light. It requires only the chemical gradient between the vent fluid and the surrounding water, and as long as the vent is active, that gradient persists. The bacteria sustain the tube worms. The clams sustain themselves by housing chemosynthetic bacteria in their gills. The crabs eat the bacteria and whatever else they can find. The entire community is energetically uncoupled from the sun, above the vents and above the floor and above the abyssal plain. The midnight zone extends through its thousands of meters of cold water. The fish and squid and crustaceans and jellies that inhabit it move through it without the benefit of sunlight, without the benefit of vent heat. They are the middle world, the water column between the productive surface and the scattered resources of the floor. Some of them migrate. They rise through the midnight zone each night into the mesopelagic and even into the shallower water above, following the signal of whatever light remains in the upper ocean, feeding in the comparatively food-rich water closer to the surface before descending again before dawn. This migration runs vertically through hundreds of meters of water column every day, driven not by seasons or weather, but by light. The rising and falling of a daily vertical migration tracks the boundary of detectable illumination with a precision that reflects how sharp and consistent that boundary is. The organisms doing it have no other navigation to trust. In total darkness, without the compass of a gradient from bright to dim, the midnight zone would be featureless to any instrument that depends on reflected light. A shadow requires a light source. A silhouette requires something to be silhouetted against. A visual field requires photons arriving from somewhere, and in the bathypelagic, no photons arrive from anywhere but the organisms themselves. This is the condition the animals of the midnight zone have inherited, a world where nothing outside casts light, where the only photons available are the ones their own bodies or the bodies nearby emit. It is not a world that evolved to receive light from a star. It is a world that evolved to work in the total absence of one, and to use whatever other sources of photons it could produce or find. That the animals of this world produce light at all is in the arithmetic of energy and expenditure. Making light costs something. It costs the chemical energy stored in a molecule that must be built, maintained, and either regenerated or replaced. In an environment where food is scarce and arrives slowly, expenditures need justification in the currency of survival. A bioluminescent organ that costs energy to produce and energy to operate represents a bet, a wager that what the light accomplishes is worth what the light consumes. The dark ocean is full of animals who have made that bet in many different forms for many different purposes. Their bioluminescent structures are evidence not of extravagance, but of tight biological accounting. When an organism that lives at 3,000 meters and eats every few days maintains a row of light-producing cells along its belly, those cells are not decorative. They are earning their cost. The accounting is precise. Below the reach of sunlight, the geometry of visibility inverts. On the surface, the brightest light comes from directly above, from the sky, from the reflected glare of the sun off the water, and the dark is below. In sunlit water, a fish that wants to avoid being seen from above makes itself blend with the darkness under it. A fish that wants to avoid being seen from below makes itself blend with the light above it. But in the midnight zone, the background is uniform and lightless in every direction. There is no sky direction and no floor direction in terms of ambient illumination, because there is no ambient illumination. The only light available to see by is the light something else is producing. This means that producing light, or not producing it, is itself a form of visibility control. An organism that glows is visible. An organism that does not glow is not. An organism that glows only when it needs to be visible and goes dark when it needs to be invisible is using light the way a lantern is used in a dark cave, not as a background condition, but as a deliberate signal with a specific context. The evolutionary pressure to refine this use, to control its timing, its direction, its intensity, its pattern, is immense in a world where being seen by the wrong thing at the wrong moment is fatal, and being seen by the right thing at the right moment is the only way to find a mate or lure a meal. The water accepts all of this with the same stillness it has maintained for hundreds of millions of years. It is cold and dark and still, and it is full of light that belongs to no star. Consider the pressure and what it does to the body. A human being taken suddenly from the surface to 1,000 meters would experience the failure of every air-filled space in the body, the lungs collapsing, the sinuses compressing, the small bones of the inner ear pushed inward by forces beyond what the structures were built to resist. The engineering of the deep-sea body is the engineering of an organism that has been worked on by that pressure for long enough that the working shows all the way down to the molecular scale. Bone density adjusts, swim bladder contents change with depth. Enzymes fold into configurations that stay stable under loads the surface equivalent would not survive. The fish that lives at 2,000 meters is not a surface fish that has been squeezed. It is a fish that pressure has assembled over deep time into a pressure-stable form. The outside of it looks like a fish. The inside of it looks like a solution. The cold contributes to this in ways that are less visible but equally pervasive. At temperatures near 2 degrees, the membranes of cells that are tuned for surface temperatures would solidify. The lipid bilayers would lose their fluidity, and the proteins embedded in them would stop moving as they need to. Deep sea organisms maintain cell membrane fluidity at low temperatures by incorporating different lipid compositions, higher proportions of unsaturated fatty acids, whose bent molecular chains pack more loosely and remain fluid where a saturated chain would lock rigid. This is not a choice. It is the slow filter of selection removing, generation by generation, any individual whose membranes failed in the cold, until what remains are the lineages whose lipid chemistry happened to work. The body of a deep-sea fish is a record of every pressure and temperature challenge its ancestors survived. Within the mesopelagic, in the twilight, where a trace of surface light still arrives, the physics of visibility is more complex. Here, there is still a downwelling light. It comes from above, and it has a spectrum strongly peaked in the blue-green wavelengths that water transmits best, stripped of red and orange and most of the yellow by the journey through 200 meters of ocean. This faint, spectral remnant of sunlight creates a gradient. Looking upward from 500 meters, an organism with sensitive enough eyes would see a faint blue-gray luminosity overhead. Looking down would reveal nothing. An animal hovering at 500 meters and silhouetted against that faint overhead light would be visible from below, a dark shape against a lighter background. To an upward-looking predator, it would be readable. This is a problem that the mesopelagic has been solving for a long time, and the solution it has converged on most widely is light. Not reflected light, not borrowed light, generated light, produced by the organism itself, tuned to match the faint downwelling spectrum from above. When the intensity and color of the ventral light output matches the intensity and color of the light arriving from above, the silhouette dissolves. From below, there is no shape where the fish was, only the same faint blue-green field that extends in every direction. This is counter-illumination, and it is practiced by hatchetfish, by certain lanternfishes, by midwater squid, by a range of crustaceans distributed through the twilight zone. It is optical camouflage built not from the absence of light, but from its precise production. It requires the organism to regulate its output in response to changing ambient light. Darker when the water above is darker, brighter on clear days when more sunlight penetrates, adjusting for depth and angle and the shifting spectrum of whatever the surface is doing, far overhead. In the midnight zone below, there is no downwelling light to match. The counter-illumination strategy becomes pointless in a world where the background is uniformly black. But the uses of light do not disappear. They change. They become more varied, more specific, more dependent on the particular circumstances of each organism's life in the dark. The logic of the mesopelagic is the logic of camouflage and silhouette. The logic of the bathopelagic is something else. The logic of a world where light itself is the signal, where producing it or receiving it, or mimicking it, or concealing it, represents the full range of strategies available to an organism that can neither see by nor hide from ambient light, because there is none. The vent community at the bottom of all of this is exempt from these optical calculations in a different way. Its organisms are not bioluminescent in the way of midwater fish and squid. They do not produce light for signaling or camouflage. The vent is chemosynthetic, not photosynthetic, and the animals clustered around it have no relationship with light at all. They do not need to be seen. They do not need to see. The tube worm, Riftia pachyptilla, which can grow to more than 6 feet in length and live in dense thickets around the vent chimneys, has no eyes. The clams and mussels that filter feed in the warm water zone near the vent opening have simplified visual systems or none at all. They navigate their world through chemical gradients, through the sensing of hydrogen sulfide and temperature and flow, through a sensory register that has no need for photons. In the vent community, the universe of light simply does not exist as a relevant dimension of experience. The vast dark water above them contains all the creatures for whom it does. Somewhere in the upper reach of the midnight zone, near the boundary with the mesopelagic, the first faint bioluminescent traces appear from above, the downward migrating animals carrying their ventral photophores with them as they sink, the slow snows of dead organic matter, occasionally carrying bacteria that still glow faintly as they decompose. This is the soft ceiling of the dark zone, lit not by any sun, but by whatever the organisms above it and within it are generating themselves. Below that, the darkness deepens toward the floor, in the way that darkness can only deepen when there is truly nothing adding to it. The floor receives what the water column above it has allowed to pass through. Sediment covers it, in some places to depths of hundreds of meters. The slow accumulation of millennia, each layer laid down in a year, that has long since become anonymous geology. In other places, the floor is bare rock, lava from the spreading centers, cooling so slowly in the cold water that it forms pillow shapes, rounded lobes of basalt, still warm at the center when they harden at the edge. Between the plains and the ridges and the canyons and the trenches, the floor of the deep ocean is more varied in its topography than most of what lies above sea level. Mountain ranges longer than the Andes run the length of the mid-ocean ridges, and trenches cut through the crust to depths that swallow the peaks of any surface mountain. The Mariana Trench drops to nearly 7 miles below sea level. The pressure there is more than 1,000 atmospheres. Life has been found there too, in the sediment, in the trenches, in places where the conditions would have seemed, before the finding, to be past any reasonable biological limit. The limit, it turns out, is flexible. The molecules of life, proteins, lipids, nucleic acids, can be assembled into configurations stable enough to function at 1,000 atmospheres and 2 degrees Celsius, if the selection pressure for that stability has been applied long enough and the variation available for selection to act on has included the right mutations. The deep sea is not a hostile environment for the organisms shaped by it. It is their normal environment. The ocean floor is where they are from. The water above them and the darkness in it is simply the world. What persists through all of this, through the pressure and the cold, through the scarcity and the stillness, through the vast distance from any sunlit surface, is the fact of life carrying on, not struggling to carry on, not surviving despite the conditions. Carrying on because the conditions are what the life here was built for, in the long, patient way that selection builds things, not by intending a result, but by preserving whatever happened to work and discarding whatever did not, generation after generation, in water that has been cold and dark for longer than there have been vertebrates anywhere on the planet. At 2,500 meters, near a vent along the Galapagos Rift, where the hot mineral fluid rises through cold water in plumes, a tube worm extends its red plume into the current. The plume is red because it is full of hemoglobin, and the hemoglobin is there not to carry oxygen, though it does, but also to bind hydrogen sulfide and transport it inward to the chemosynthetic bacteria living in a specialized tissue called the trophosome, which fills most of the worm's body cavity. The worm has no mouth, it has no gut. It is in its adult form entirely dependent on its internal bacteria for nutrition, and they are entirely dependent on the chemical energy of the vent and the oxygen the worm carries down to them. The partnership is precise. The vent is the engine, the worm is the delivery system, the bacteria are the kitchen. Around the tube worm, in the warm current rising from the vent, other organisms move or stay still. The water nearest the vent chimney is too hot for most of them. The shrimp and crabs that cluster at a small distance have found the thermal margin where the vent's warmth diffuses into the surrounding cold water and the gradient produces the conditions they can tolerate. They press as close as the heat allows. The warmth itself is energy, and energy here is the rarest resource. To be near the vent is to be near the source. Beyond the warm zone, the abyssal plain extends away in every direction in the dark, cool, unhurried, and vast. The marine snow falls through it at whatever rate the surface has permitted that week, that month, that season. A sea cucumber at the edge of the vent community moves across the sediment on tube feet at a pace that makes the slow movement of glaciers seem hurried. It consumes the sediment it passes through, extracting whatever organic matter the particles contain, and leaves the clean sediment behind in a thin trail. The trail will persist in the undisturbed silt for days. It is a record of something moving through a world where almost nothing moves. This is the bottom of the world that bioluminescence inhabits. This is the dark that shaped it, the 2 degrees of cold, the 100 atmospheres or more, the total absence of sunlight, the silence of water that does not storm, the scarce food, the long, slow generations of animals refined to work here and nowhere else. The light that the organisms of the midnight zone produce did not arise because the ocean wanted it or worked toward it. It arose because in certain populations, in certain lineages, in certain moments of the deep past, a particular chemistry that produced light happened to be useful. It helped hide something. It helped something find a mate. It helped something lure a meal close enough to catch. It persisted because it worked, and it was refined because the refinements worked better, and it was reinvented because the dark ocean poses the same problems in the same way, wherever it exists, and chemistry is not infinite. The vent at the Galapagos Rift knows nothing of all this. It has been pushing hot mineral water into the cold ocean for thousands of years, and the organisms around it have been sustaining themselves on that heat since before any human being thought to look for them. The water above the vent is as dark as the water 500 miles away, as dark as the water over the abyssal plain, as dark as the water at the base of any ocean trench. The darkness does not vary. It does not thin toward the floor or thicken toward the surface. Below 1,000 meters, the dark is simply the dark, complete, ancient, and still, with no requirement for anything to be different, and no memory of the light above. In that water, in the cold and the pressure, and the endless falling of marine snow from a surface it will never reach, life has arranged itself into 10,000 solutions to the problem of surviving in the dark. Some of those solutions glow. They glow steadily, or in pulses, or only when touched, or only in the presence of enough of their own kind to warrant the expense. The glow crosses the dark water for a few centimeters or a few meters, and something sees it or nothing does. And in either case, the water closes back around it and the dark resumes, untroubled as it has always been, cold and wide and permanent, and full of living things that have learned to carry their own light. In the black water below a kilometer of sea, a small light hangs still. It does not flicker, it does not sweep or search. It holds its position in a column of water, so cold and so dark that nothing in the world above would recognize it as a place at all. The light is blue, faintly blue, the color of the hour before dawn, compressed into a point no wider than a fingertip. Around it, in every direction, the water is absolute. No sun reaches here, no moon, no diffuse gray of a cloudy sky, no phosphorescence of a surface wave. There is only this light, steady as a coal, and the enormous darkness surrounding it, and whatever slow, enormous shape breathes behind it in the black. The light belongs to a fish, or rather the light belongs to what grows from the fish, a modified spine of the dorsal fin that has over a vast span of time migrated forward until it curves above the fish's head like a fishing rod above a still pond, and terminates in a fleshy knob called the esca. The esca is hollow, and the light inside it is not the fish's at all. Packed inside that knob of flesh, in chambers the fish grew specifically to contain them, are living bacteria, not a few millions, dense enough that their population within the esca represents one of the more concentrated communities of microorganisms in the entire ocean. Each bacterium is small and unremarkable in isolation. In open water, bacteria of this kind do not glow. They drift in the dark and divide and go about their lives in the same lightless chemistry as everything else down here. But when they find themselves packed together beyond a certain density, when the molecules they have been quietly releasing into their immediate surroundings build up to a concentration that registers as enough, something in their gene expression changes. The light-producing chemistry switches on. It does not switch on gradually. It reaches a threshold, and then it is there. So the esca glows. The fish whose body grew the esca never produced a single photon of it. The anglerfish, for this is the ceratioid anglerfish, the creature at the end of the lure, is a carrier, an architect, a host. Its body built the chamber and maintains it, supplies it with oxygen and the raw materials the bacteria need, and in return it inherits the light that the bacteria, by their own inner arithmetic, decide to make. The esca hangs in the darkness ahead of the anglerfish's jaws. The jaws are enormous, wider than the body in some species, lined with long translucent teeth that curve inward like bent wire. The fish drifts without swimming, barely moving, using almost no energy in an environment where food can be days or weeks between encounters. The lure holds still, the light holds still, and somewhere in the water column nearby, above, below, to the side, something smaller is moving toward it. Whatever moves toward the light moves toward it, because in an ocean where almost nothing produces any illumination at all, a steady blue point of light is worth investigating. It may be a smaller animal with its own photophores. It may be the bioluminescent flash of a disturbed dinoflagellate, which in these depths means something edible was just startled. The anglerfish waits, the bacteria glow on, and the darkness that was the problem becomes, through this patient collaboration between a fish and its microscopic tenants, the solution. Below the anglerfish's drift, deeper in the column, the water is colder still. Down here, the pressure has been increasing with every meter of descent, and the creatures that live in this range carry that pressure in the molecular architecture of their bodies, in the shape of their enzymes, in the chemistry that keeps their proteins stable. Most of them are small. Many of them are dark, and a number of them carry light, not borrowed from bacteria, but built entirely within their own tissue, in organs evolved across their lineages for exactly this purpose. These organs are called photophores. The word means light bearer, and in the animals of the deep ocean, it describes a structure of considerable complexity, not a simple patch of glowing tissue, but an assembly of lens, mirror, pigment layer, and light-producing cells, all arranged so that the light produced inside the organ goes exactly where the animal needs it. Some photophores point down, some point sideways, some flash rapidly, and some hold a steady emission. And in the fish of the deep mesopelagic zone, the distribution of photophores across the body is not random. It follows a pattern that, once understood, reveals the particular problem the animal is solving. The viperfish is long and thin and hung with rows of light. It lives in the midnight zone, somewhere between 200 and 1400 meters depth, depending on the hour. Because the viperfish does not simply live at one depth, it moves. Each day, each real day, measured by a sun, the viperfish has never seen and will never see. It makes a vertical migration that would be scaled to a human body, something like running from sea level to the summit of a mountain and back again twice in 24 hours. It rises toward the surface at night, where prey is denser. It descends before dawn, retreating back into the cold and the dark. And throughout this movement, the rows of photophores along its belly are doing something specific. The photophores of the viperfish's ventral surface emit a faint blue-green light. Faint but not absent, and the wavelength they emit is very close to the wavelength of the last most attenuated traces of sunlight that filter down into the upper reaches of the mesopelagic, the faint blue-green shimmer that is all that remains of the sun at 400 or 500 meters. A predator sitting below the viperfish and looking upward would, without the photophores, see the fish's silhouette against that faint residual light. The shadow would be a signal, an edge, a shape, a movement. But with the photophores running, the underside of the fish emits just enough light at just the right wavelength to fill in that shadow. The silhouette dissolves. The fish becomes, to a predator below, a small and invisible interruption in the ambient glow of a fading sky. This is not something the viperfish chooses or adjusts consciously. The light organs respond to the ambient light levels around them as the fish rises or descends. As the background light brightens or fades, the photophores modulate their output to match it. The system holds the balance across the full range of the migration, maintaining the erasure at each depth with a precision that has been refined, generation by generation, across an enormous span of time. Along its modified first dorsal fin ray, the viperfish also carries a lure, different from the anglerfish's esca in construction and origin, but serving a related purpose. The tip of the ray glows, and with the fish can arch the ray forward over its head, positioning the light in front of its mouth, where the teeth wait. The teeth of the viperfish are long enough, and the lower jaw hinged wide enough, that the fish cannot fully close its mouth around them. They protrude forward when the jaw is open, a row of curved transparent spines in front of the lure, in front of the light. When something comes to investigate the glow, it arrives at the worst possible geometry. There is something about the viperfish's existence that takes a long time to settle. It spends its life moving between two layers of a lightless ocean, lit from below by its own photophores, and from above by the dying edge of a day it never directly experiences. The sun matters to the viperfish, determines its movements, calibrates its camouflage, dictates the schedule of its hunting, but only as an absence, only as the presence or lack of a faint blue-green residue in the water above. The viperfish knows the sun the way a creature might know the weather in a country it has never visited, by the effects that arrive, faint and second-hand, through a great depth of something cold. The water at night, where the mesopelagic touches the shallower coastal depths, moves differently, where currents carry warmer water against a coastline, where the seafloor rises and the water column compresses, where the chemistry changes and the prey concentrates. Certain animals gather in numbers that make the water itself seem alive. Off the coast of Japan, in a bay where cold and warm waters meet in spring, a particular squid makes a migration that has been watched and documented and still resists easy summary. The firefly squid is small, a body the length of a finger, tentacles adding a little more. It lives at depth during the day, but as darkness reaches the surface, millions of individuals begin rising, not hundreds, not thousands. The numbers involved in the spring spawning aggregation in Toyama Bay have been estimated in the hundreds of millions. The bay, in late March and April, appears at night to be lit from below. Each firefly squid carries photophores across its mantle and along its arms and tentacles. The distribution is dense and complex, and the light produced is a vivid blue. When the squid flash their photophores, which they do in patterns and sequences that vary with context, the individual pulses last a fraction of a second and can be repeated rapidly. From above, the surface of the bay during a peak spawning event appears to move with light, waves of blue flashing traveling through the water as the aggregation shifts and turns. Aircraft have observed these events. The glow is visible for considerable distances. The photophores of the firefly squid are not simply lights that turn on and off. They are structured organs, and the squid has more variety of visual pigment than most cephalopods, 3 distinct types, where many squid have only one. A richer visual capacity for a creature that produces and reads complex light signals is not a coincidence. The evidence points toward a system of communication, not language, nothing as structured as language, but a system of light-based signals that carry information about identity, reproductive state, and possibly position within the aggregation. The squid can see each other's lights and respond to them. The flash patterns are not random noise. They are, in some sense, that researchers are still working to characterize, a conversation conducted in blue. The spawning females come to the shallows and release their eggs, and the light continues through the night, and then the tide moves and the water stills, and by morning the bay is ordinary again. The squid that survived the night return to depth. The eggs drift, and the bay, for another day, holds its ordinary appearance, cold and gray and unremarkable from the surface, with no visible record of what happened below it in the dark. There is another light in the night ocean that belongs to no single organism large enough to see. It belongs to a category of life so small that 1 million individuals fit comfortably in a teaspoon of seawater, and yet collectively they have been observed from orbit. Dinoflagellates are single-celled organisms, plentiful enough in certain coastal and open ocean conditions to form what are called blooms, concentrations, so dense that they visibly change the color of the water in daylight, giving it a red-brown tone that has in some regions given a name to the phenomenon. At night, the same cells produce light. The light comes from structures inside the cell called scintillons. Each scintillon is a small membrane-bound compartment containing the molecules needed for a bioluminescent reaction, a form of luciferins specific to dinoflagellates and the enzyme that processes it. The reaction sits ready, loaded, waiting. What triggers it is mechanical disturbance, a wave breaking, a fish moving through the water, a hand or an oar or the bow of a ship. When the cell membrane is deformed by pressure or turbulence, ions move across the membrane of the scintillon, the chemistry inside shifts, and the reaction fires. The flash lasts approximately a tenth of a second. Then it is over, and the chemistry reloads, and the cell waits again. In a dense bloom, the effect of a breaking wave is to trigger millions of these flashes simultaneously, each cell responding to the same mechanical disturbance, each releasing its brief pulse of blue-green light, the collective output bright enough to make the foam of a wave glow, to make the wake of a boat trace a luminous line across the water, to make the handprint of a swimmer hang blue and briefly visible in the dark surface of the sea. The light is cold, it contains no heat. The dinoflagellate cell is not damaged by it, is not warmed by it, does not suffer for producing it. The chemistry has been arranged across a very long time so that almost all the energy of the reaction goes into light rather than into heat, a nearly complete conversion that still exceeds what human engineering can achieve in most applications. The cell spends this energy in the moment of disturbance and then rebuilds its supply of luciferin during the following day, ready for the next night. Why a single-celled organism would spend energy producing light is a question with a specific and rather elegant answer. The flash is not for the dinoflagellate's benefit in any direct sense. It is, the best current evidence suggests, an alarm, but an alarm directed not at the creature attacking the cell, but at something else entirely. A copepod grazing on a dinoflagellate causes a flash. That flash attracts the attention of a small fish. The small fish redirects toward the copepod. The copepod, which was the predator in the original encounter, becomes prey. The dinoflagellate's flash summons a predator of its own predator, and the grazing stops. This does not work every time. The fish does not always notice. The copepod sometimes escapes anyway. But across the statistical span of millions of cells and billions of encounters, the cells that flash when disturbed fare slightly better than cells that do not. And that small difference, compounded across generations long enough to fill geological time, is sufficient. The flash persists. The chemistry that produces it persists. And on warm nights and bays where the water has concentrated enough cells, the whole living surface of the sea becomes a light-producing network, responsive to every disturbance, every breath of wind, every passing animal, a vast and decentralized alarm system stitched through billions of tiny individual lives. A ship moving through such a sea at night leaves a trail behind it that fades slowly, blue-white and churning at the stern, narrowing to a thin line and then to nothing as the water stills. Each point of light in that trail was a cell. Each flash was a tenth of a second of chemistry, followed by a return to waiting. The trail is made of individual lives, responding one by one, and their collective response looks from the ship's deck like something continuous, like a single luminous wake, when in fact it is an aggregate of discrete, tiny, precisely managed events. The light that travelers have seen on night crossings, the light that has made sailors record strange observations in logs that were later filed away as weather phenomena, the light that has glowed on beaches where waves break in summer, a significant portion of it comes from this source. From organisms too small to see individually, producing light in response to touch in a chemistry refined across hundreds of millions of years, for a reason that has nothing to do with beauty and everything to do with survival. Far from the coastlines, in water that is neither shallow enough for kelp nor deep enough for the permanent midnight zone, the open mesopelagic ocean holds its own population of light-producing life. Much of it is small. Many of its inhabitants are translucent, glassy, built to be seen through rather than around. And among the oldest lineages of bioluminescent animals in this zone are the jellyfish, medusae drifting in slow pulses through the middle water, their bells contracting and releasing in a rhythm as old as the animal itself. One species in particular carries a light that has traveled further than the ocean it came from, off the coast of the Pacific Northwest, in the cold water that moves between the open sea and the sounds and straits of the Washington coast. A jellyfish drifts. The bell is translucent, nearly transparent, in fact, a quality that in water at this depth allows the animal to remain largely invisible to anything looking at it from the side. The bell is perhaps a few inches across. The margin of the bell is fringed with small structures, approximately 100 of them arranged in a ring around the edge, that produce light. The light is green. This is Aquaria victoria, the crystal jelly. It lives along the surface and in the upper depths of the Pacific coast, feeds on other small zooplankton, and has a lifespan measured in weeks or months. Nothing in its ordinary existence marks it as unusual. Its bell folds and opens in the rhythm common to all metaeus. It drifts where the current takes it. It does not migrate vertically. It does not produce a lure or maintain bacterial colonies or flash as an alarm. The green glow at the margin of its bell is simply present, a faint, steady luminescence visible in the dark water through the near-transparent walls of the organism, as if the edge of the bell has been traced in a color slightly different from the ocean around it. The chemistry behind that glow involves two proteins working in sequence. The first is contained in the photogenic cells along the bell's margin and responds to calcium ions. When calcium is present in sufficient concentration, the protein undergoes a reaction and releases blue light. In isolation, the system produces blue, but the crystal jelly's photogenic organs contain a second protein, distributed alongside the first, which absorbs that blue light and re-emits it at a longer wavelength. The blue becomes green. The organism has built, within its own margin, a two-stage light conversion process, a molecular relay, in which the output of one reaction becomes the input of another, and the final color that emerges is different from the color that the chemistry alone would produce. The two proteins are distinct from each other, each with its own structure and function. The first, which fires in response to calcium and releases blue light, was given the name equorin. The second, which absorbs the blue and reissues it as green, was given the name green fluorescent protein. In the summer of 1961, a researcher named Osamu Shimomura arrived at the Friday Harbor Laboratories, a marine biology station on San Juan Island in the waters of Puget Sound. He had come specifically to study the light of Aquaria victoria and to isolate from the jellyfish's tissue whatever it was that made the glow. The work required jellyfish, enormous numbers of jellyfish. Shimomura and his colleagues collected them by hand from the docks, using dip nets, in the early morning hours when the animals concentrated near the surface. They cut the light-producing margins from the bells and processed the tissue. In the first summer, they collected and processed tens of thousands of individual jellyfish. In subsequent seasons, the number grew higher still. Estimates across the years of the project run to over 800,000 animals, each contributing a small amount of its photogenic margin to the accumulating extract. From that extract, Shimomura eventually isolated aquarin and identified its calcium-triggered mechanism, but alongside it was the green fluorescent protein, quieter in the biochemistry of the live animal, responsible only for the color shift, and initially set aside as secondary to the primary discovery. It would be decades before another researcher recognized that this secondary protein, the one that absorbed blue light and released it as green, had a property that had no parallel anywhere else in biochemistry at the time. It could fluoresce inside a living cell without any additional chemistry, without a substrate, without the cellular machinery of the host organism needing to do anything to activate it. Other ways of tagging proteins in living cells required complex preparation, or damaging conditions, or external reagents. Green fluorescent protein needed only light. Aim a particular wavelength of blue light at a cell that had been made to express green fluorescent protein fused to any protein of interest, and the green protein would glow, and the protein of interest would glow with it, tracing its movement through the cell in real time, through the living cell's own membrane, without killing it. This is not described here for what it led to. That belongs to another part of this story. What matters here in the water is the object itself, a jellyfish, translucent, drifting in cold water off the coast of Washington on a dark morning in any of the summers between 1961 and whenever Shimomura last collected, the bell folding and opening, the green light at its margin, faint enough that it would take a dark room to see properly, steady enough that it had been noticed by people working on the dock late at night. For generations, the jellyfish did not know its light was being observed. It had no knowledge of the dip nets, of the tens of thousands of nights it took to accumulate enough extract, of the cold laboratory where its proteins were slowly separated from each other by centrifuge and column and patient chemical inquiry. It continued to drift in the current and fold its bell and glow faintly green at its edge, because that is what Equoria victoria does in the cold water of the Pacific coast, as it has done across an enormous span of time that predates every institution built to study it. The bell of the crystal jelly is nearly weightless in water. The contraction of the muscle along the bell's rim expels water downward, and the animal moves upward, slowly, a few centimeters per pulse. Then it relaxes, and the bell opens, and the animal settles slightly. Open, close, open, close. The rhythm is unhurried. The water around it is dark and very cold. The green light at the bell's margin persists through each contraction and each opening, unaffected by the movement, steady in a way that the motion of the animal does not disturb. At the margin, in the tissue of the photogenic organs, the two proteins sit together in the same cell. The aquarin waits for calcium. The green fluorescent protein waits for blue light. The cell lives its cellular life, maintaining its membrane, managing its chemistry, doing the small, unremarkable work of staying alive that every cell performs continuously and without fanfare. And in the dark water of a Pacific Sound, the green light that these two proteins together produce traces the edge of the bell like a signature written in a color the ocean almost never uses. There is something about the convergence of these five organisms, the anglerfish with its bacterial lamp, the firefly squid with its blue aggregation, the dinoflagellate with its alarm of 10 milliseconds, the viperfish with its counterfeit sky, and the crystal jelly with its two-stage green conversion that asks the mind to slow down and consider the breadth of what one chemistry can accomplish. The underlying reaction in all of them involves a light-emitting molecule and an enzyme that processes it, oxygen mediating the transaction, and the energy released going into a photon of visible light rather than into heat. The details differ. The molecules are different across lineages. The enzymes share no meaningful ancestry. The structures that house the reaction range from single-celled organelles to complex multi-layered organs evolved entirely within vertebrate tissue. But the output is the same: cold light in the dark made by living tissue without burning anything. Nature arrived at this chemistry not once but dozens of times. Each arrival was independent. Each evolved from whatever raw materials were available in that lineage's biochemistry, fatty acid processing enzymes, repurposed, calcium-sensitive proteins co-opted, metabolic reactions redirected. The deep ocean did not require that life produce light, but life, encountering the deep ocean's darkness, found its way to light again and again through different paths, the way water finds different routes down the same hillside, but arrives in the same valley. In the dark water where none of this is visible from the surface, the anglerfish holds its lure still. The bacteria inside the esca count their own numbers in the accumulated molecules of their shared space, and glow because the count is high enough. The viperfish moves upward through 1,000 meters of dark water toward the faint and vanishing blue-green of a sunset it will never see, its belly lit to match the sky it is rising toward, its lure pulsing slowly at the tip of its modified fin. The firefly squid, in their hundreds of millions, press toward the surface of a bay in spring, each one carrying the blue vocabulary of a communication older than human language. The dinoflagellates drift in the upper ocean, loaded and waiting, their scintillans ready, their alarm already primed by the physics of water moving against a small and patient cell. And somewhere along the coast of a cold Pacific Sound, a crystal jelly folds and opens and folds again, its margin green, its proteins at work inside the cold tissue, its light available to the dark water and to any instrument patient and precise enough to come and find it. The glow at the margin of the bell is steady. It has been steady for a very long time. The ocean around it has been dark for longer than the organism has existed, and the organism has existed for longer than it has been observed, and it will drift and fold and glow in the cold water of the Pacific long after the last note written about it has yellowed and faded in whatever archive holds it. The light does not need a witness. The chemistry does not require an audience. In the dark water below the reach of sunlight, in the cold and the pressure and the long, slow downward drift of marine snow from the surface above, the cells that carry light make it anyway, in the dark, as they have always done, as the ocean moves around them and the years accumulate and the current carries them on. A row of cells runs along the underside of a small fish, each one carrying a tiny point of light. The fish hangs in the water at around 400 feet down, where the last trace of sunlight arrives from above as a faint blue-green wash, diffuse and directionless, barely enough to cast a shadow. From below, a predator might be looking upward, and what the predator sees, or rather, what it does not see, is the silhouette of a fish. The row of lights along the fish's belly has been calibrated over an immensity of time to match the precise color and intensity of the downwelling light above. The shadow that would betray a living body simply does not form. The fish is there, and the fish is invisible, and the mechanism that makes it so is a row of cells that glow. This is counter-illumination. It is a solution so geometrically exact that it requires very little description beyond the geometry itself. There is light above, there is a predator below. Between them, an animal must either avoid detection or accept being seen. The row of ventral photophores, the small specialized light organs along the belly, does not work by accident. Through long refinement across many generations, those cells came to produce light that matches the residual glow of the surface. Not brighter, not dimmer, not a different color, matched the way a paint chip is matched to a wall, except the matching here was done by survival across millions of predator-prey encounters, and the animals that matched poorly were eaten, and the animals that matched well persisted and had descendants. What remains is a living instrument of optical precision, suspended in 400 feet of darkening water, erasing itself from the view of anything looking up. Hatchetfish carry this adaptation with particular refinement. Their bodies are flattened side to side, deep and narrow, shaped something like the blade of an axe, and their ventral photophores face straight downward in a long continuous array. The light they produce sits in the blue-green part of the spectrum, the same part that sunlight concentrates into as it filters through seawater, losing its reds and oranges and yellows within the first 100 feet of descent. At the depth where hatchetfish live, the remaining light is so faint and so narrowly blue that it is almost beyond perception, but the photophores match it with great accuracy, and the hatchetfish's silhouette dissolves into the faint luminous ceiling above it. A predator looking upward through the water finds not a fish-shaped shadow, but an unbroken continuation of the dim and featureless glow. The fish is there. The evidence of the fish is not. Many species of lanternfish use a version of the same approach. Lanternfish are among the most numerous vertebrates on the planet by count, small and silver-scaled, and numerous beyond easy imagining in the mesopelagic waters of every ocean, and their photophores are arranged in species-specific patterns that serve multiple purposes, counter-illumination among them. The precise arrangement differs from species to species in ways that appear to carry information, which is something worth holding gently in mind, because the same light that hides one creature from a predator may simultaneously be readable to another creature of the same kind, and this layering of function inside a single behavior is one of the deep characteristics of bioluminescence throughout the ocean. Midwater squid carry photophores on their mantle and arms, and some species can adjust the output of their ventral organs in real time, dimming or brightening as the ambient light above changes with depth or time of day. The control is active, not passive. The animal is not simply glowing at a fixed intensity. Something within the animal's nervous system is comparing the light hitting the upward-facing surface of its body with the light being produced by its downward-facing photophores, and adjusting the output to maintain the match. The result is a continuously updated optical illusion, and the fact that this calibration happens inside a living creature moving through a column of water that is itself changing by the minute is one of those quiet biological achievements that requires a moment's pause before continuing. The same darkness that makes camouflage necessary also makes predation difficult. Food is sparse in the deep ocean. The animals that live there have evolved around scarcity in ways that are visible in their bodies: enormous jaws, expandable stomachs, teeth designed to hold rather than chew, every calorie of a meal accounted for. In this environment, luring prey toward the predator rather than chasing it is an energetically efficient strategy, and bioluminescence makes luring possible. A light in absolute darkness is one of the most attention-commanding things that can exist. Any organism that has evolved to associate light with food, with the faint bioluminescence of the prey it normally hunts, will be drawn toward a similar light, even if the source is not what it seems. Dragonfish exploit this with a structure that extends from their chins, a long and flexible barbel tipped with a photophore and a surrounding array of secondary light organs that flash in patterns. The pattern is not random, or rather, it appears not to be random. The flashing sequences of some dragonfish barbel tips resemble, at least in general character, the bioluminescent displays of the small crustaceans and copepods that other midwater fish hunt. Whether a passing prey fish is drawn toward a dragonfish lure because the lure resembles something familiar, or simply because any light in the dark is worth investigating, is a question that remains open. What is clear is that the lure works. The dragonfish waits, the prey comes toward the light, and the dragonfish, which may be nearly invisible itself. Some species are so deeply pigmented that they absorb almost all light that strikes them, takes the meal with very little expenditure of energy. The deep-sea anglerfish uses a similar principle with a different anatomy. The lure sits not below the chin, but above the head, a modified fin ray that curves forward over the mouth like a fishing rod, and at its tip glows the esca, the small bulb of tissue lit from within by bacteria. The anglerfish does not produce this light. The light is produced by microorganisms living inside a specialized chamber within the esca, and the fish provides the bacteria with a warm and nutrient-rich home in exchange for their glow. The economy of this arrangement is worth noting. The fish does not need to synthesize the chemical components of bioluminescence on its own, which requires metabolic investment. It simply houses the organisms that already know how to glow, and it points them forward into the darkness. The lure hangs steady. Other animals, drawn toward the point of light in the blackness, come within reach of the jaws. The bacteria glow on without any apparent awareness of the role they are playing. Light production of this kind, where a host animal carries symbiotic bioluminescent bacteria inside a dedicated organ, is a different relationship than the intrinsic bioluminescence of animals that produce their own light through their own biochemistry. Both strategies exist in the ocean, and they solve the same problem through different means. An animal that produces its own light through its own enzymes has full control over when and how the light is produced, but it must maintain the molecular machinery to do so. An animal that houses luminescent bacteria delegates the production and accepts the dependency on a living partner that must be cultivated and maintained. Neither strategy is better. Both persist because both work, in different bodies, in different ecological contexts, across many separate evolutionary histories. Some deep-sea squid use their photophores in a different way still, not to hide, not to lure, but to mimic. The arrangement of light organs along their bodies and the timing of their activation can produce patterns that resemble the bioluminescent signature of other organisms. A squid hunting smaller fish that are themselves attracted to certain bioluminescent displays might produce a pattern that resembles whatever those smaller fish are looking for. The predator becomes, in a sense, a counterfeit version of the prey's own prey, and the deception operates entirely in light, in the darkness, without color or shape, only the timing and spacing of photons, arranged to suggest something that is not there. This is a mechanism whose precise function is still being worked out, and the best current picture is that photophore mimicry operates in at least some species of midwater cephalopods as a form of aggressive luring, though the evidence is incomplete and the ocean keeps its own record. There is something in the structure of these deceptions that rewards slow attention. Each one operates on the same principle. A signal that evolved in one context is exploited in another. A prey animal evolved to recognize and approach a bioluminescent display because that display indicated food. A predator evolved to reproduce that display, or something close enough to it, because the proximity of the attracted prey provides a meal. The prey's recognition system, which served it in one context, becomes a vulnerability in another. And because the deception happens in darkness, at depth, in cold water, with no witnesses except the participants, the precision required is low. Close enough is close enough. If the prey animal has no way to check its assumptions before coming within reach of the jaws. Bioluminescence as communication carries a different register than bioluminescence as camouflage or predation. When light carries information between members of the same species, rather than deceiving members of other species, the logic of the signal shifts. The receiver is the same kind of animal as the sender. The signal is being read by a visual system shaped by the same evolutionary pressures that shaped the signal itself. And what the receiver is reading, in many deep-sea fish, appears to include information about identity, sex, and reproductive condition. In a number of species of deep-sea fish, the arrangement and patterning of photophores differs between males and females in consistent and species-specific ways. The differences are not subtle. In some species, males carry photophores in locations where females have none, or the reverse. In others, the flashing pattern during active display differs between sexes in timing, intensity, or the direction in which the light is produced. These differences constitute a form of visual language, readable at short range through absolute darkness, and the best current picture is that they function in mate recognition and assessment. The dark ocean offers no other channel for this kind of information exchange at the distances and light levels involved. Sound can travel, but many of these small midwater fish have limited acoustic signaling. Color is impossible where there is no ambient light. Shape can only be read at distances where a predator can also see. Light produced from within the body, at a wavelength tuned to the sensitivity of the receiver's visual system, may be the only reliable channel available. The evolutionary logic of sexual dimorphism in photophore patterning is consistent with what is observed in bioluminescent organisms in other contexts. Light does not carry just one kind of meaning. A single photophore can contribute to counter-illumination in one orientation, to species recognition in another, and perhaps to something else entirely in a third context. The function depends on what is receiving the signal and how that receiver interprets it. This flexibility, the same physical structure serving multiple purposes depending on context, is one of the reasons bioluminescence has persisted and diversified so widely in ocean animals. A set of light organs that already exists for camouflage can be co-opted over time to carry additional information if the information proves useful, and it costs nothing in terms of the hardware already in place. A dinoflagellate is a single cell. It has no nervous system, no sensory organs, no capacity to assess the situation it finds itself in and decide on a course of action. And yet when it is mechanically disturbed, when the water around it is suddenly compressed and released by a passing body, a sweeping appendage, the bow wave of a copod or a fish, it flashes. The flash lasts about 1/10 of a second. It is blue, and it is bright enough in the darkness of the open ocean at night to be visible to any eye in the vicinity. For a long time, the flash was described as a side effect, something that happened because the cellular machinery was triggered, with no particular consequence worth examining. The current picture is more interesting. In experimental conditions, populations of dinoflagellates embedded in the path of grazing copods have been observed to flash when the copods disturb the water around them, and the flashes appear to attract the attention of small fish in the vicinity. The fish redirect toward the point of light. The copod, in the act of eating the dinoflagellate, has made itself visible to a predator that was not looking for it a moment before, and the copod is eaten. This is the burglar alarm hypothesis. The single-celled organism cannot run. It cannot fight. It has no way to discourage a copod from eating it, except to make the copod's presence known to something that will discourage the copod by eating it instead. The flash in this reading is not a signal to the predator that is attacking it. The dinoflagellate has no way to choose its signals and no way to address them to specific receivers. The flash is simply light, released in darkness, visible to whatever happens to be paying attention. that a fish might be paying attention, and that the fish might respond to the light by approaching, and that the copod might be caught in that approach, is a consequence that requires no intention on the part of the dinoflagellate. The mechanism is simple. The effect is a small, cold piece of ecological logic operating at the scale of a cell, repeating across billions of individual encounters in the open water, every night, in every ocean. The copod that ate the dinoflagellate is now the prey. The fish that saw the flash is now the predator. And the dinoflagellate that flashed, or what remains of it, has, by the fact of its chemistry, altered the outcome of the interaction between two other organisms, not through any quality of mind or intention, through the simple fact that it contained the molecules that produce light when compressed, and that it was compressed, and that in the dark ocean, light draws attention. The flash is a consequence. The consequences of the flash are a chain. The chain runs through bodies that have no knowledge of each other, and its links are made of photons. There is another kind of relationship between light and living organisms in the deep ocean, one that operates not between predator and prey, or between members of the same species, but between host and symbiont, between a large organism and the microorganisms it carries inside a dedicated structure within its own tissue. The light organs of certain fish and squid are not just photophores, they are ecosystems. Small, sealed, internally ventilated chambers lined with reflective tissue and capped with a translucent window, and inside them, maintained at careful density, live colonies of bioluminescent bacteria. The bacteria produce light continuously when conditions are right, but in open seawater, the bacteria are scattered and dim, too few of each other's kind to activate the genes that control light production in coordinated fashion. Inside the light organ, the conditions are different. The bacteria are packed tightly together. They have been growing and reproducing inside the organ, and their population has been climbing. As the population grows, each bacterial cell releases small signaling molecules into the fluid around it. When those molecules accumulate to a threshold concentration, when enough cells are present that their chemical signals pile up dense enough to trigger a response, the light-producing genes switch on across the population more or less simultaneously. This is quorum sensing. It is a form of collective counting, performed by organisms that have no nervous system and no awareness of anything beyond their immediate chemical environment. Each cell is simply releasing molecules and responding to molecules, but the collective effect is a population that measures its own size and changes its behavior at the threshold. Below the threshold, the bacteria are dark. Above the threshold, they glow. And inside a fish's light organ, the threshold is reached and maintained. And the organ glows with a steady light that the fish uses for whatever purposes its anatomy has arranged the organ to serve: camouflage, luring, species recognition, or some combination that varies with the depth and the moment and the particular history of that lineage. The fish controls the light in various ways, despite not producing it. Some fish have a pigmented shutter, a flap of skin that can be drawn across the organ to block the light entirely. Some have reflective backing tissue that can be angled to direct the light in one direction or another. Some can alter the blood flow to the organ, changing the oxygen availability and thus the intensity of the bacterial glow. The host animal does not tell the bacteria what to do. It manages the context in which the bacteria find themselves, adjusting the conditions until the output matches what the animal's own behavior requires. The bacteria glow because the quorum is maintained. The host maintains the quorum through the architecture and management of the organ. The result is a light source that the host can modulate without controlling at the molecular level, a delegated production line managed at arm's length, with consequences that serve the organism that runs the organ. What is quiet in this arrangement is how ancient it is. The partnership between light organ fish and bioluminescent bacteria is not a recent invention. It is a relationship that has been maintained across many millions of years in multiple separate fish lineages, each of which developed a light organ independently, and each of which came to be colonized by bacteria that were already present in the seawater around them. The bacteria did not come from elsewhere. They were drawn from the ocean's own microbial population, selected because they could survive and multiply inside the organ. And because they glowed. The fish did not go looking for luminescent bacteria specifically. It evolved an organ that created conditions favorable to bacteria generally, and the luminescent bacteria thrived there because they were already present and because the organ suited them. What the two partners exchanged was not a conscious bargain. It was a set of conditions that turned out to be mutually sustaining, and that has remained mutually sustaining through a length of time that makes human history look very brief. Consider what happens along a stretch of open ocean on a dark, calm night at the surface where the water meets the air. A wave builds slowly from the action of a distant wind and moves shoreward across the flat black water. Inside the wave, the water is in motion, compressed and released, rolled forward, stirred against itself, and in the water are millions of single-celled organisms, each one small beyond easy visualization, each one carrying within it the molecular machinery that produces light when disturbed. The wave reaches the shore and breaks, and the breaking water glows, not continuously, not steadily, but in brief pulses, each one 1/10 of a second long, each one blue, each one produced by a single cell in the moment of its disturbance. The whole breaking wave is a collection of individual flashes, each triggered by the same physical event, each brief and local, and together they produce something that resembles a continuous sheet of cold blue fire running along the sand. This is what a dinoflagellate bloom looks like from the shore. The glow is not one light. It is millions of lights happening at once in the same space, each one independent of the others, each one triggered by the mechanical disruption of breaking water, rather than by any awareness of what the others are doing. The coordination is not social, it is physical, the same force moving through the same body of water, reaching each cell at nearly the same moment, and each cell responding in the same way. The result looks like something designed, but the design is in the cells themselves, each one identical in its response, and the wave provides the simultaneity. Seen from above, from an aircraft, or from low orbit, the glow of a large dinoflagellate bloom in breaking surf can be resolved from a great distance. The blue light is faint by most measures, but the darkness of the ocean at night is absolute, and faint light in an absolute dark is visible far beyond what it would be in daylight. The bloom marks its own presence across a surface that is otherwise featureless and black, and the billions of cells producing it have no awareness of the scale of what they are collectively making visible. Each one is only responding to the water moving against it. Each one flashes and goes dark. The pattern they make together belongs to a scale none of them can perceive. There is one more function of bioluminescence that sits apart from camouflage, from predation, from communication, and from the ecological logic of the burglar alarm, and that is the function that operates within symbiotic relationships at the level of the bacterial community itself. The quorum sensing mechanism that controls bacterial bioluminescence is not unique to light production. It is a general regulatory system used by many bacteria to coordinate collective behaviors, the production of toxins, the formation of biofilms, the release of enzymes, and the fact that it controls light production in luminescent species is, in an evolutionary sense, an application of an existing tool to a new problem. What this means is that bioluminescence in bacteria is not simply a trait that evolved in isolation. It is connected to a broader framework of collective bacterial behavior, a molecular system of social coordination among organisms that are simultaneously some of the simplest and most numerous forms of life in the ocean. When a light organ fills with glowing bacteria, what is visible from outside is a steady point of light. What is happening inside is a dense community of microorganisms, each monitoring its chemical environment, each releasing and receiving signals, each responding to the accumulated signal of its neighbors, all of them crossing a threshold together and illuminating from within. The light is a byproduct of this collective state. It is also the whole reason the organ exists. And it is also, at the same time, a bacterial behavior that is entirely indifferent to the fish around it. The bacteria would glow at quorum density in a glass vessel just as readily as they glow inside a light organ, because the quorum sensing system does not know or care what surrounds it. This indifference is worth holding. The bacteria are not serving the fish. They are responding to their own population density with a behavior that evolved for reasons unrelated to fish, in ocean waters where no fish light organ contained them, and the light organ came later, built by a lineage of fish that found a use for the thing the bacteria were already doing. The fish built the conditions, the bacteria filled them. The light emerged from the meeting of those two trajectories, each one independent, each one ancient, and neither one aimed at the outcome that resulted. In the midnight zone of the ocean, below 3,000 feet, where no sunlight has ever reached, and where the water has been cold and dark for longer than most life on this planet has existed, the functions of bioluminescence overlap and layer over each other in ways that are still being untangled. An animal at that depth may use light to hide from predators below, while simultaneously using a different set of light organs to signal to potential mates above, while the bacteria in a third set of organs glow because the quorum has been reached and they have no other response available. All of this is happening in the same body, in the same moment, in the same column of cold black water. The light serves predator avoidance, reproduction, and microbial population dynamics simultaneously, not because any organism planned it that way, but because each function was added to what already existed over long separate intervals of time, and the combination that survives is the combination that was compatible with surviving. A hatchetfish moves slowly through the water at 400 feet down, its photophores dimly lit against the faint blue above it, its silhouette dissolved into the uniform glow of the surface light. It is looking for food. Something below it may be looking for it. Its photophores are adjusted continuously to match the available downwelling light, a calibration so habitual that the fish does nothing to perform it. The light simply is what it needs to be, and the fish moves through the water in the way that keeps it fed and unfound for another day. Somewhere below the hatchetfish, beyond the last residue of downwelling light, in water so cold it has barely changed temperature in 100,000 years, something pulses. A brief blue flash, 10 milliseconds wide, released from a single cell, disturbed by a passing current. It travels a short distance through the water, weakening as it goes, and arrives at nothing in particular, no eye to receive it, no organism primed to respond. It dissolves into the dark. Another flash follows nearby, from another cell, also triggered by the passing current, and another, not coordinated, not aimed, each one a consequence of a cell doing what its chemistry requires. When the water moves, the dark absorbs them, one after another, small and cold and brief, and the ocean around them continues as it has continued, steady, unhurried, indifferent to the lights going on and off within it, vast and dark and full of small living things, each one carrying in its cells the chemistry of light, waiting quietly for the particular disturbance that will bring it briefly into view. The quorum gathers inside a light organ somewhere in the same water. The bacteria multiply, the signaling molecules accumulate in the fluid around them, each cell adding its share, the concentration climbing slowly toward the threshold. Not there yet. The organ is dark. The fish moves through the cold water, and the bacteria inside it go on with the only thing they do, growing, dividing, releasing their small molecules into the enclosed space, counting in the only way available to them, which is to let the molecules pile up until there are enough. The threshold approaches. In the water outside, another dinoflagellate flashes and goes dark. A copod sweeps through a cloud of cells, disturbing the water, triggering a brief scatter of blue light across a small volume of open ocean, and then going on, unaware of what it has made visible, moving through the darkness, the way everything moves through the darkness. At that depth, slowly, steadily, with no light of its own and no expectation of any. The threshold inside the light organ is reached, and the bacteria glow, quietly, steadily, without event, without announcement, without any signal that something has changed. Only the even and continuing blue-green light of a bacterial community large enough to illuminate itself, held inside a chamber built by a living body, pointed outward into a dark that has been waiting for it. In the summer of 1934, a steel sphere 48 inches across hung on a cable above the Atlantic Ocean, off the coast of Bermuda, and then went down. It weighed nearly 2.5 tons. Its walls were 1.5 inches thick. Inside, a single small window of fused quartz looked outward into water that was growing darker with every foot of descent. 2 men sat inside that sphere. There was room for little else, and one of them pressed his face to the glass and watched the light go. The quartz window was barely 8 inches across. Through it, the naturalist William Beebe could see the color of the sea changing as the cable let out above him. At the surface, it had been green, carrying in it all the turbulence and life of shallow water. At 30 feet, it had shifted toward blue. At 200 feet, the blue had deepened past anything a painter would find on a palette, a blue so dense it seemed almost structural, as if the water had become a solid thing. And then, somewhere below that, the blue thinned. It did not turn to gray. It did not turn to black the way a room goes dark when a lamp is switched off. It dissolved the way a sound dissolves at the edge of hearing, not absent exactly, not yet, but present only as a quality of the water itself, a cold pressure of fading that grew absolute so gradually that Beebe could not say precisely when the last light ended and the true dark began. At 923 feet, the cable stopped. The bathysphere hung in water that had not been touched by sunlight in any meaningful sense since long before any human was alive to wonder about it. The temperature of the water outside the steel wall was close to 40 degrees Fahrenheit. The pressure against every square inch of that hull was greater than the weight of a full-grown man. Beebe sat in the dark of the sphere's interior, lit only by a small light he had brought with him, and he looked through the quartz window, and he waited to see what lived out there. What he saw had no names. That was one of the first things that struck him, not the darkness, not the pressure, not the cold transmitted faintly through the hull, but the absence of language for what he was watching. Forms drifted past the window that did not correspond to anything in the catalogs of known deep-sea life, shapes that carried their own light. A line of pale blue-green luminescence describing a curve that passed through the beam of his exterior lamp and then continued into the dark beyond it, something larger and dimly lit from within, moving with a slow steadiness that suggested no urgency at all. A flash, sudden and brief, at the edge of visibility, and then nothing. He wrote descriptions into a microphone connected to a telephone cable running up to the ship above, and a secretary on deck transcribed them, and later those descriptions would fill pages in his published accounts, strained always toward precision and always falling short of what he had actually seen. He was the first scientist to observe deep-sea bioluminescence directly, in place, in the water where it had evolved and persisted, and found its purposes across hundreds of millions of years. Every specimen that had been brought to the surface before him had arrived damaged, compressed by the sudden change in pressure, bleached of color, dark the light already extinguished in the time it took the net to come up. What Beebe saw through that 8-inch window was something that had never been seen before by human eyes, and he had nothing to measure it against and no instrument capable of capturing it. He had only his face at the glass and the cold, dark water on the other side of it, and whatever moved through that water carrying its own faint and steady glow. He came back to the surface. The cable lifted the sphere. The blue deepened and then thinned, and then the green of shallower water returned, and the surface broke over the porthole, and the hatch was unsealed, and the two men climbed out into the Bermuda sun. Beebe spent years trying to put into exact words what he had seen down there, and the words were never quite adequate, and he knew it. What he had witnessed was a world that existed entirely on its own terms, lit by its own means, indifferent to the sun above, and the ships crossing on the surface, and the single small steel sphere that had descended into it for a few hours in 1934 and then gone away again. The sphere itself was retired eventually. It sits now in a museum, its quartz window replaced, its surface bearing the marks of its 115 dives. The ocean it descended into is still there, and still dark, and still full of organisms producing light, and the vast majority of what Beebe watched through that window that summer afternoon has never been given a formal scientific name. For decades after Beebe, the deep ocean was approached mostly from above. Nets were lowered and retrieved, samples were brought up. The specimens that arrived at the surface were studied, cataloged, and described, their photophores measured, their anatomies mapped, their evolutionary relationships inferred from what remained after the pressure change had had its way with them. It was useful work. It built the foundations of what is known. But a pressed flower is not a living one, and a dead lanternfish brought up from 500 meters in a trawl net does not show anyone how a living lanternfish moves through the water at 500 meters, or what its lights are doing as it moves, or how the animals around it respond to those lights. The living behavior was gone. What remained was structure without context, the instrument without any record of the music it had played. The difficulty was physical and fundamental. To observe something at depth without disturbing it, an instrument had to reach that depth without carrying the light that would change the behavior of everything around it. Most deep-sea animals are sensitive to visible light, not only to the bioluminescence of their neighbors and prey and predators, but to any light at all that arrives from directions it should not arrive from. A lamp mounted on a submersible floods a field of view with light that no organism at that depth has ever encountered in the course of its evolution. The animals that don't flee go still. The interactions that would have occurred in the dark do not occur. The observer, carrying light, is measuring the absence of what the observer wants to measure, and the measurements feel like data but are shaped entirely by the act of looking. The solution, when it came, was simple in principle and required years of engineering to make real. A researcher named Edith Witter, working at the Harbor Branch Oceanographic Institution and later at the Ocean Research and Conservation Association, understood the problem clearly. The light had to be invisible, not dimmed, not filtered to a wavelength less alarming to the animals. Invisible, outside the range of the visual systems of the organisms being observed. Most deep-sea animals have visual pigments tuned to blue and blue-green wavelengths, the colors that carry farthest in clear deep water, and the colors produced most commonly by bioluminescent organisms. What they do not have, in the great majority of cases, is sensitivity to far-red wavelengths. Red light, to a creature whose eye has never had reason to develop receptors for it, is no different from no light at all. Witter designed a camera system around that fact. The eye in the sea, as it came to be known, used red light, light invisible to the organisms around it, to illuminate a field of view. The camera that recorded that field was sensitive to red wavelengths in a way no animal at that depth was. The result was an instrument that could see without being seen. It could be lowered to the seafloor and left there recording, while the animals around it went about the business of living as if nothing unusual had arrived in their darkness at all. No alarm, no freeze, no flight response, just the ordinary behavior of the deep ocean floor, proceeding in the ordinary dark, captured at length by a device that was effectively transparent to everything it watched. The system was deployed at many depths and many locations over the years, producing footage that shifted the understanding of deep-sea behavior in ways that catalog data from trawled specimens simply could not. Animals were observed interacting, feeding behaviors were recorded, bioluminescent signals were captured passing between individuals of the same species, pulses and flashes, and steady glows trading across short distances of dark water, carrying whatever it is they carry between creatures whose entire social world is conducted in light they themselves produce. In one deployment, the camera was baited with a device that mimicked the bioluminescent distress pattern of a small deep-sea jellyfish, a rotating LED array set to produce a specific pattern of blue flashes that Witter had identified in the field as a signal produced by prey organisms under attack, a call that appears to summon larger predators to the location of the disturbance. The eye in the sea recorded what came to investigate. What came, moving into the frame in 2004 at a depth of nearly 3,000 feet in the Gulf of Mexico, was a giant squid. It was the first footage ever recorded of a giant squid moving freely in its natural habitat, not a dead specimen hauled up from a trawl net, not a partial body recovered from the stomach of a sperm whale, a living animal arriving out of the dark, its arms moving with a slow, deliberate articulation, its body bearing the faint iridescence of a creature that had never needed to be seen and had never been. It examined the bait. It was not alarmed. It departed into the dark again, unhurried. The footage ran for a few minutes, and then the squid was gone, and the seafloor was dark again, the way it had been before the camera arrived, the way it would be after the camera was retrieved. The giant squid had been theorized, described from fragments, drawn from sailors' accounts for centuries. Its existence had been deduced from the sucker scars it left on sperm whale skin, circular marks, some nearly 2 inches across, pressed into the hide of animals that dove to great depths in search of food. Everything known about it before that footage had been inferred from evidence gathered at surfaces, the surface of the sea when specimens washed ashore, the surface of a whale's skin, the contents of a stomach opened on a research vessel's deck. The animal itself had always remained below, in the water where it lived, moving through a darkness that no instrument had been able to enter without announcing itself. The camera had entered without announcing itself. That was the whole of the achievement, and it was not small. In the same decades that observation technology was advancing downward into the ocean, the molecular tools developed partly from deep-sea biology were advancing inward into the cell, into the nucleus, into the signaling pathways that govern how living tissue grows and divides, and occasionally fails. The story of how those tools came to exist begins, like much of the science of bioluminescence, in shallow cold water, in a very different kind of patient work. Near the Friday Harbor Laboratories on San Juan Island, off the coast of Washington State, in the early 1960s, a Japanese chemist named Osamu Shimomura was collecting jellyfish. The species was Icoria victoria, a small translucent animal, not striking in appearance, drifting along the surface of the Pacific in dense seasonal aggregations near the shore. What Shimomura was interested in was the ring of tissue at the margin of the jellyfish's bell, a fringe of roughly 100 small photogenic organs that produced, under the right conditions, a faint greenish luminescence visible through the animal's near-transparent body. He wanted to know what produced the light, not in ecological terms, in molecular terms. What substance, what chemical reaction, what protein or small molecule was responsible for the photons coming out of that tissue. The work required large numbers of jellyfish. Shimomura and his colleagues collected them by hand in buckets from the water near the laboratory, cutting the light-producing margins from the bells and processing them immediately to preserve whatever was in that tissue before it degraded. Over several seasons, the number of jellyfish processed reached tens of thousands. The tissue extracts were refined and tested. Two distinct molecular components of the light-producing system were eventually isolated. One was a photoprotein, a protein that bound a small light-emitting molecule and held it ready to react. In the presence of calcium ions, the reaction ran and the protein emitted blue light. This protein was named Icorian after the species it came from. The other component was something different, and at the time, not immediately understood. It absorbed the blue light produced by Icorian and emitted green light in its place. Under the conditions inside a living jellyfish, the two proteins worked in close proximity, the blue output of one becoming the input of the other. The final color shifted from blue to the soft green, visible at the bell's margin. This second protein was the one that would eventually carry the science of cellular imaging into territory that had been unreachable before it. It was called green fluorescent protein. For some years after its isolation, it remained a laboratory curiosity, characterized, published, set aside. What it was, chemically, was understood. What it could do for biology, beyond the biology of bioluminescence, was not yet clear. The clarity came later, and from a different direction. A molecular biologist named Martin Chelfie recognized that the gene encoding the protein could in principle be fused to the genes encoding other proteins inside living cells, causing those other proteins to carry the green fluorescent marker wherever they went inside the cell. No injection of dye, no fixed and dead tissue, a living cell expressing green fluorescent protein as part of its ordinary life, with that fluorescence attached to whatever molecule the researcher wanted to track. The first demonstration was in a transparent worm. The cells in which the fused gene was expressed glowed green under ultraviolet light, and the worm was alive, and the locations of those cells in the worm's body could be seen directly and mapped and followed over time as the worm grew and developed. It worked exactly as predicted. The tool was real. A biochemist named Roger Chean took it further still. The green fluorescent protein had a specific molecular structure responsible for its color, and that structure could be altered. Specific amino acids changed, the geometry of the light-emitting core adjusted to shift the emission wavelength. Seeing engineered variants of the protein that emitted not green, but blue, cyan, yellow, and eventually red. Multiple variants could be expressed simultaneously in the same cell, each attached to a different protein of interest, each glowing in a distinct color, visible as separate signals under the right illumination. A cell with multiple fluorescent proteins expressed inside it became organisms under the microscope, a small landscape of distinguishable lights, each color marking a different molecule, moving through the cell's interior on its own path, interacting with other colored markers in patterns that could now be watched in real time in living tissue. Shimamura, Chalfie, and Cheen shared the 2008 Nobel Prize in Chemistry for the discovery and development of green fluorescent protein and its applications. The prize acknowledged a lineage of work that ran from buckets of jellyfish in cold Pacific water to the interior of cancer cells viewed under a confocal microscope in a hospital research laboratory. The green that had evolved over vast spans of time to shift the emission of a bioluminescent photoprotein in the bell of a translucent coastal jellyfish had become, by a chain of patient steps, the same green that a researcher watched moving through the nucleus of a dividing human cell, or along the branching extensions of a neuron, or accumulating at the membrane of a cell, responding to a signal from outside. The molecular mechanism was the same. The context was entirely different. The light came from the same cold ocean it had always come from, translated now into the interior of a different kind of living thing. None of this could have been predicted from the jellyfish. No one collecting Aquaria Victoria in buckets in 1961 was thinking about cellular imaging, the connection between a small coastal invertebrate and the ability to watch a cancer-related protein migrate through the nucleus of a human cell in real time was not a connection anyone was looking for. It was found by working carefully on a small and specific question: what makes this animal glow? And following the molecular answer wherever it led, which turned out to be further than anyone had anticipated. That is one version of how this science tends to move, not from problem to solution, but from patient observation outward in directions that cannot be predicted in advance. By the late 1990s and into the first decade of this century, the pace of deep-sea observation had increased in ways that would have been difficult to imagine from inside a steel sphere on a cable in 1934. Remotely operated vehicles, unmanned, tethered, equipped with high-definition cameras and articulated arms and banks of lights in calibrated wavelengths, had made it possible to reach depths of 2 miles or more and remain there for hours, transmitting video in real time to researchers on the surface vessel. Ambari, the Monterey Bay Aquarium Research Institute on the California coast, operated vehicles capable of working at depths of nearly 13,000 feet. The footage accumulated over those years amounts to a library of deep-sea behavior that no previous generation of scientists had access to in any form. The organisms filmed in that footage included many that had never been seen alive before, species known only from trawled specimens, compressed, discolored, their bioluminescent tissues dark and collapsed, appeared for the first time in their living form, moving through the water at depth, their photophores active, their behaviors coherent and purposeful in ways no dead specimen could have revealed. Animals whose photophore arrangements had been mapped from preserved tissue turned out in footage to produce far more complex and dynamic light patterns than the static map had suggested. Species that had been classified as solitary On the basis of the low probability of two individuals arriving in the same trawl net turned out to aggregate at certain depths and certain times, their bioluminescent signals passing between individuals in patterns consistent with active communication. The technology also made systematic survey possible at scales that net-based sampling could never approach. An ROV moving slowly through a column of water at a known depth and speed, recording continuously, could produce a reliable estimate of the density and diversity of organisms present in that column. In a way that a trawl net, which catches different organisms with different efficiencies depending on their speed and behavior, could not. When surveys using this approach were compiled across many locations and depths as part of the Census of Marine Life, a program running from 2000 to 2010 and involving more than 2,700 researchers working across 80 nations, the picture that emerged was striking in its proportions. In the Mesopelagic Ocean, the water column between roughly 600 and 3,000 feet, the zone where sunlight fades and finally ends, more than 3/4 of the observable organisms in many survey regions were found to produce bioluminescence. Not all organisms at those depths produce light, but of the ones that could be observed, moving freely through the water, the majority carried photophores or other light-producing structures and used them. The deep ocean was not a place where bioluminescence was an occasional adaptation, a specialized trick found in a few unusual lineages. It was the dominant mode of existence at those depths, light produced by the organisms themselves being in a very real sense the primary light of that world. The census also produced an estimate of how much of the deep sea had been formally surveyed and how much remained outside any systematic human observation. The covered areas, mapped against the total volume of ocean available at mesopelagic and bathypelagic depths, formed a pattern that would be modest by any standard of coverage, narrow tracks through an enormous space, scattered by geography and funding and the limited operational range of the vessels and vehicles that had done the work. Hundreds of thousands of deep-sea species, the census researchers estimated, remain formally undescribed. Not undetected, there is enough indirect evidence from the diversity of organisms found in even the most intensively sampled regions to know that the catalog of known deep-sea life is a small fraction of the actual total, simply unnamed, unstudied. Their bioluminescent signals, if they produce them, unrecorded. The ocean below the reach of sunlight covers most of the floor of this planet. Its volume exceeds the combined volume of every other habitat on Earth, and the greater part of what lives in it has not been seen, has not been named, and will not be named in any near future that can be reasonably imagined. The instruments exist in principle to reach most of those depths. The organisms are there, producing light in the dark, as they have been since long before the first human eye existed to look for them. The gap between the instruments and the ocean is one of time and resources and the slow, patient accumulation of survey effort that this kind of science requires. Nothing more dramatic than that, and nothing less enormous. William Beebe lived until 1962. He never descended in the bathysphere again after 1934. In his later years, he wrote that the deep ocean was the largest unexplored territory on the planet, and that the few hours he had spent looking through that quartz window had convinced him that what remained below the reach of any cable he could lower was stranger and more various than anything the surface world had prepared him to imagine. He was right about the strangeness. He was right about the variety, and the territory he named as unexplored remains, by most measures of exploration, as unknown as it was when he said it. The steel sphere he descended in was 48 inches across. The ocean he descended into is at its greatest depth, nearly 7 miles. The difference in those two scales is not a metaphor or a rhetorical gesture. It is a plain statement of the relationship between the instrument and the thing the instrument entered. One small sphere on a cable in the dark, and around it, in every direction but up, more water than the sphere could contain in any number of hypothetical lifetimes of descending. What Beebe saw through the window was real and true, and documented as carefully as the tools available to him allowed. What he could not see was everything else, the water extending away from the window on all sides, in the dark, carrying its population of luminescent organisms, going about their lives in the ordinary darkness that had always been their medium and their world. That water is still there. The organisms in it are still producing their cold light. The light travels a short distance through the water and then fades, the way all light fades in the absence of a surface to reflect from, and the dark resumes, and the organisms move on, and no record is made of any of it. There is something in that continuity worth resting against. The light has been produced in that water for a very long time, longer by many orders of magnitude than the time humans have been alive to wonder about it, longer than the time there have been eyes of any kind at those depths to see it or respond to it. The chemical reaction runs in the cold water, the substrate meets the enzyme, the electron drops to its lower energy state, and a photon of blue-green light is released into the dark. This has happened billions of times today alone, in water that no instrument has reached. It will happen again tomorrow, and the day after, in the same cold dark with the same chemical precision, whether or not any record is ever made of it. The photon travels a few meters and is absorbed. The water is dark again. The animal that produced it moves on, or rests, or feeds, or is fed upon, continuing the life it has always led in the place it has always led it. And the water around it holds in every direction, at every depth, the same steady biological activity, the same faint and various luminescence moving through the dark, the same cold chemistry running in the tissues of creatures no catalog has yet reached, the same old light produced by the same old reaction in the same old dark, continuing quietly, as it always has, needing nothing from above. The ocean is deep and cold, and mostly dark and mostly unnamed. Its volume is very large. The organisms in it are very many,